Hello, and welcome to this presentation on a Galaxy-based multi-omic informatics hub for cancer researchers for the ITCR project. This project is focused on building flexible and accessible informatics resources for multi-omics research. But what is multi-omics? Multi-omics is the integration of different omic data types such as genomics, transcriptomics, proteomics, and metabolomics. Each omics method contributes to a more complete understanding of the biological system under study. For this project, we will focus on mass spectrometry-based data, including proteomics, wherein protein molecules present in a biological sample are studied, and metabolomics, wherein molecules acting as reactants and products in enzymatic reactions are studied. When mass spectrometry data are integrated with genomic information, DNA sequences can be linked with expressed proteins and metabolites, indicators of biological function, providing a more complete snapshot of the underlying mechanisms that cause cancer. Methods integrating different omic data include proteogenomics and metaproteomics. DNA sequence data alone provides only a look at what might be happening in cancer development and progression. Adding proteomic or metabolomic information provides a more complete look at what is actually happening in the system, helping to develop more accurate methods for detecting and treating cancer. For each of these omic data types, there are a bevy of bioinformatic tools available. By combining the results of one tool to another, we can get a more complete and nuanced profile of a biological system. However, putting this into practice can be overwhelming, since operating these software tools separately is time-consuming and ultimately not feasible. Fortunately, there exists a solution to address these issues by bringing all of these tools together in an accessible, user-friendly, web-based bioinformatics platform, aptly named Galaxy. In the Galaxy interface, the web browser is divided up into 3 panes, one showing available software tools. Here, tools from different omic types can be easily accessed for multi-omic analysis. A center pane for viewing operations such as tools, datasets, and visualizations. And lastly, a panel for your history, which contains all the data inputs and tool results generated from your analysis. As an open source project, Galaxy ensures shareability between researchers, which can facilitate multi-omics research. Users can choose to share or publish their analysis history like such. However, histories are generally limited to past analyses. To reuse an analysis on other data, users can use workflows, which are essentially blueprints that direct how multiple tools handle input data. They can be generated from an existing history like this, or can be built from scratch or edited in the workflow editor based on your research needs. Much like histories, workflows can be shared for use with the community. Now that we've shown you Galaxy, let's show you some multi-omic applications, such as metabolomics. Metabolomics is the study of small molecules, or metabolites, in an organism. Organisms interconvert metabolites in a series of chemical reactions catalyzed by enzymes within metabolic pathways. Researchers can use metabolomics to study how an organism's metabolome is affected, which is an indicator of biochemical activity of the system. For instance, plants could be grown under different light conditions to study how metabolic pathways respond. Or, scientists may also look for a metabolic signature or fingerprint of a disease or stress. Cancer is known to affect this fingerprint, and these can act as markers of cancer progression and treatment. A metabolite signature could be useful for early detection of cancer. Our metabolomics focus is on developing tools and data processing workflows in Galaxy, which quantify and determine metabolites of highest interest within a specific sample. Our metabolomics software development can be followed on the Hegman Labs GitHub page, and much like proteomics.usegalaxy.eu shown earlier, we are working on making these tools available on accessible resources such as metabolomics.usegalaxy.eu. Proteogenomics is a multi-omic approach, integrating genomics, transcriptomics, and proteomics approaches to identify proteins that are novel or variant. Genes that make up the genome carry information for expressing specific proteins. Any change in the nucleotide sequence of a gene can lead to a change in the sequence and function of a protein. Codons, which are formed by 3 nucleotides, code for specific amino acids that are the building blocks of proteins. However, even a single change in the codon sequence can lead to changes in the amino acids in a polypeptide chain. This change of amino acids causes expression of a variant protein, which may cause abnormal biological function leading to various disease conditions, such as cancer. Biological studies focusing on genome or DNA sequences do indicate the possibility of causal mechanisms for any disease condition, but they do not depict the complete picture. Integrating proteomics data, which directly detects the expressed variant proteins, with the genomics and transcriptomic approaches helps reveal the mechanisms of cancer development and progression. Integration of many software and tools is required to perform a multi-omics analysis like proteogenomics. There are many useful genomics, transcriptomics, and proteomics software tools available. However, there is a need for a unified platform that can enable their integration. The Galaxy platform is well suited to act as this unified platform. We have developed proteogenomic tools to visualize results and to empower cancer researchers to interpret results and form new hypotheses. For example, the Multi-omics Visualization Platform, or MVP, is a visualization and validation tool for proteogenomics results. MVP can be launched from a proteogenomics analysis history in Galaxy and can help in the validation of the data quality of identified variant proteins. It also enables viewing the protein sequence in the context of the genes which encode it using the Integrated Genomics Viewer, or IGV. The GalaxyP tool in Galaxy is a proteogenomic extension of the web-based cancer-related analysis of variants toolkit, also known as GalaxyP. GalaxyP retrieves cancer and functional annotation from genomic, transcriptomic, and proteomic data and helps researchers understand the possible impact of variants. Results can be visualized with a compatible interactive plugin allowing exploration of the data via circus plots, 3D protein models, protein domain lollipop diagrams, and molecular network analysis using the Index software infrastructure. Finally, the QuantiP tool allows integration of transcriptomic and proteomic data by enabling systematic comparison between abundances of RNA transcripts and proteins. These outputs are visualized and interpreted through various interactive plots, facilitating the identification of genes that may have post-transcriptional regulation. This reveals potential new mechanisms underlying cancer. Microbiome research, or the research of the effect of microorganisms present in a host or an environment, has provided deep insights into how it affects human health, disease, and also the environment. Recent studies have also shown the effects and interactions of human microbiota with its host to influence cancer physiology. Multi-omics research for microbiome involves using metagenomics, which looks at microbial DNA, metatranscriptomics, which is concerned with microbial RNA, and metaproteomics, which examines microbial proteins for microbiome research. Metaproteomics involves mass spectrometry analysis of proteins expressed from microorganisms within a host, such as a human. This provides a look at molecules from microorganisms which may interact with the host and contribute to health and disease such as cancer. When combined with metagenomics, which can identify which microorganisms are present, metaproteomics gives a more complete picture of molecular contributors and possible functions from microorganisms, such as bacteria in cancer. A successful metaproteomics workflow involves many data processing steps, such as database generation, peptide spectral matching, taxonomy analysis, functional analysis, and quantitation. This requires software from disparate sources to integrate metagenomic sequencing data with metaproteomics data. We have developed workflows for processing and integrating metagenomic and metaproteomic data. Galaxy provides a powerful environment to implement and combine these software. Visit z.umn.edu/metaproteomics gateway for more information. We are also developing new tools to help interpret metaproteomics data. For example, we have developed a tool called MetaQuantum which quantifies proteins expressed by microorganisms and offers novel visual outputs that enables users to explore the proteins expressed by specific organisms and how these may contribute to cancer biology. Workflows for metatranscriptomics analysis have also been implemented in Galaxy. This work was done in collaboration with the European Galaxy community. Our ultimate goal is to develop and implement tools and workflows within Galaxy that can enable and integrate metagenomics, metatranscriptomics, and metaproteomics studies in the field of cancer microbiome research. We believe that a multi-omics approach at the host and the microbiome level would offer a more complete picture of how microbiomes interact with their hosts. The GalaxyP team has been involved in many activities to promote software tools and analytical workflows developed for multi-omics research. These include hands-on training workshops, presentations at conferences, online training gateways, and hosting tools on publicly accessible software repositories. One of the main activities is hands-on training workshops. We have conducted workshops at national and international conferences around the world. These include workshops on proteogenomics, metaproteomics, and metabolomics. The GalaxyP team has also presented posters and oral presentations at multiple conferences around the world. The GalaxyP team has also participated and contributed to the online community training sites such as Galaxy Training Network or GTN. Currently, the GTN has training material on Proteogenomics, Metaproteomics, and Metabolomics. We are seeking to expand the repertoire of training workflows and tools via the GTN resource. Many of the GalaxyP tools and workflows are publicly available via GitHub, Galaxy Toolshed, Docker Images, Galaxy EU, and Jetstream resources. For the last 2 years, the GalaxyP team has been disseminating workflows via scientific gateways available on the NSF cloud computing resource, Jetstream. GalaxyP tools and workflows are also available via the newly developed proteomics.usegalaxy.eu and metabolomics.usegalaxy.eu portals. These were developed in collaboration with the European Galaxy community. If you'd like to know more about GalaxyP, please go to our project website, galaxyp.org, or you can follow up on recent updates by following our account under the Twitter handle useGalaxyP. Thank you!